Hello, I'm Dr. Urano. I'm a physician scientist at Washington University School of Medicine in St. Louis, where I serve as principal investigator of the HELIOS trial and as a professor of medicine. My research and clinical work focus on Wolfram syndrome, a rare inherited neurodegenerative disorder that affects approximately 3,000 individuals in the United States and many more worldwide. Despite its rarity, Wolfram syndrome is severe, progressive, and life-limiting, making it an area of significant unmet medical need. Wolfram syndrome is most commonly caused by autosomal recessive loss-of-function pathogenic variants in the WFS1 gene. WFS1 encodes wolframin, a protein that plays a critical role in maintaining cellular homeostasis. Loss of wolframin leads to chronic cellular stress, particularly involving dysfunction of the endoplasmic reticulum and mitochondria, two systems essential for protein handling and energy production. Over time, this sustained cellular stress results in progressive cell dysfunction and cell death across multiple organ systems. Clinically, individuals typically develop insulin-requiring diabetes early in life, followed by progressive vision loss, hearing impairment, and neurologic dysfunction. As the disease advances, involvement of cerebellar, brainstem, and autonomic pathways contributes to impaired balance, coordination, swallowing, and respiratory control, leading to premature mortality. There are currently no approved disease-modifying therapies, and management remains largely supportive, highlighting the need for treatments that target the upstream mechanisms of cellular dysfunction. Given the central role of endoplasmic reticulum and mitochondrial stress in Wolfram syndrome, an investigational oral combination therapy, sodium phenylbutyrate and taurursodiol, known as PB and TURSO, was evaluated for its potential to target both pathways simultaneously in patient-derived induced pluripotent stem cell models of Wolfram syndrome. Treatment with active components led to increased WFS1 protein expression, reduced markers of endoplasmic reticulum stress and apoptosis, and improved insulin secretion, consistent with improved cellular function. These mechanistic effects were further corroborated in Wolfram syndrome mouse models, where treatment significantly delayed progression of the diabetic phenotype. Together, these convergent preclinical findings provided the biological rationale for advancing PB and TURSO into clinical evaluation and led to the initiation of the HELIOS trial. HELIOS is a single-arm, open-label phase 2 clinical trial evaluating PB and TURSO in adults with Wolfram syndrome. The primary efficacy assessment was conducted at week 24 with continued on-treatment evaluation through week 48. All 12 enrolled participants received at least one dose of study drug and were included in the safety and intent to treat analysis. One participant was later determined not to meet protocol-defined genetic eligibility criteria and was excluded from the per-protocol efficacy analysis. Accordingly, while clinical outcome analyses are presented for both the intent to treat and per-protocol populations, the per-protocol population is considered the primary dataset for efficacy analysis. Progressive beta cell dysfunction is a defining feature of Wolfram syndrome. Accordingly, the primary efficacy endpoint in HELIOS was endogenous insulin secretion, measured as C-peptide response during a mixed meal tolerance test. Shown here are the mean C-peptide response curves at baseline, week 24, and week 48 for both the intent to treat and per-protocol populations. Despite the expected natural history of progressive decline, treatment with PB and TURSO was associated with increased mean C-peptide responses at both follow-up time points. These findings suggest preservation and, in some participants, improvement of endogenous insulin secretion over 48 weeks of treatment. For individuals with Wolfram syndrome, even maintaining insulin production can be clinically meaningful. because Wolfram syndrome is usually marked by ongoing, irreversible loss of beta cell function. In addition to the observed preservation or improvement of beta cell function, reflected by improved C-peptide responses, we observed changes in glycemic measures consistent with improved metabolic control, including reductions in hemoglobin A1C and increased time spent within the target glucose range. Visual acuity was stable or improved over 48 weeks of treatment. All participants with available data were classified as responders on both participant-reported and clinician-reported global impression of change scales, indicating stabilization or improvement in overall symptom burden. Taken together, these convergent findings across objective measures and patient-reported outcomes support a multi-system signal of benefit, which is notable in a disorder characterized by relentless progression. Several limitations warrant consideration, including the small sample size and open-label single-arm design, constraints inherent to studies conducted in rare diseases such as Wolfram syndrome. PB&TURSO was generally well tolerated. Adverse events were predominantly mild to moderate and primarily gastrointestinal in nature. There were no treatment-emergent serious adverse events and no discontinuations due to adverse events during the reported period. Overall, across 48 weeks of treatment, adults with Wolfram syndrome demonstrated stabilization or improvement across multiple disease-relevant outcomes. In the context of relentlessly progressive disorder, even stabilization represents a meaningful clinical benefit. Collectively, these findings support continued clinical development of sodium phenylbutyrate and taurursodiol for Wolfram syndrome. This study is sponsored by Amylyx Pharmaceuticals. We are deeply grateful to the participants in the HELIOS trial and their families, whose commitment made this work possible. We thank the Washington University study team, collaborating investigators, and the broader Wolfram syndrome community. Thank you so much. We are so grateful.